We are delighted today to celebrate the establishment of the Dr. and Mrs. Paul J. Vignos Jr. Professorship in Rheumatology and Immunology, and the appointment of Dr. M. Edward Metoff as the inaugural chairholder. Dr. Medof came to the School of Medicine faculty 30 years ago in 1985 as an associate professor in the departments of pathology and in medicine. During his tenure, he worked with dozens of graduate students and postdoctoral fellows as a trainer in the biomedical and medical scientist training programs, and as director of the Immunology program and director of the Visual Sciences pre-doctoral and postdoctoral training program in the Department of Ophthalmology. He's quite versatile, isn't he? Dr. Medoff has also mentored younger scientists as part of a special undergraduate biology experimental immunology program. A talented clinician, Dr. Medoff is the former director of the Immunology Rheumatology Lab at the University of Chicago, where he developed a rheumatology outreach program in partnership with 5 area hospitals and provided clinical training on a rotating basis. Today, he continues to see rheumatology patients and has served as a resource for clinicians facing complicated immunological problems. Dr. Medoff's impressive body of research includes more than 200 abstracts, 165 journal articles, and more than a dozen book chapters and reviews. He also holds 12 patents and has another one pending, all related to discoveries, diagnostic tests, and therapies in the area of rheumatology, immunology, internal medicine, and cancer. Again, a broad display of immunological interests. And just this year, Dr. Medoff was appointed to lead the School of Medicine's new initiative for integrative immunobiology, an interdisciplinary hub for immunology education and research, which will be dedicated to unraveling the immune and biological processes central to human health. And we're hoping to derive funding to upgrade it to an institute. The School of Medicine has a rich and impressive history in immunology research, reaching back many decades and involving leaders in the field, beginning in the 1950s with Louis Pillemer's discovery of properdin and its role in immune response. This new initiative will build on this long history and serve as an international model for the advancement of immunobiology, bringing together experts from many divergent fields to catalyze discoveries. Dr. Medoff's broad experience in rheumatology and immunology makes him a natural fit to help us lead this new endeavor. We are able to attract and retain outstanding faculty members like Dr. Medoff because of the support and foresight of our generous philanthropists and friends. Professorships like the one we celebrate this afternoon provide an environment that allows them to excel in their work and to share their knowledge in order to advance medicine. Today, there are 110 endowed professorships within our school, positions held by faculty who are the best of the best. Nearly a quarter of these chairs have been established during the current forward-thinking campaign. So that's pretty good track record, and congratulations to our development team for that. I'm so extremely proud of the work that our endowed professors do every single day. And we're so grateful to those of you who share our vision and make professorships possible and giving us the tools we need to support these amazing colleagues. The professorship we celebrate today was made possible thanks to generous support from the Louise H. and David S. Ingalls Foundation to honor Paul A. Vignos and Edith. A quick who's who, Edith Ingalls was a daughter of Louise Harkness Ingalls and David Ingalls. She married Dr. Paul Vignos in 1946, just as his career in medicine was beginning. You've been there for all along the way, haven't you? After completing medical school here in Case Western Reserve in 1944, Dr. Vignos served in the Medical Corps of the U.S. Air Force and then completed a residency at the New York Presbyterian Hospital before returning to research at Case Western Reserve and practice at University Hospital's Case Medical Center. Dr. Vignos went on to establish the Division of Rheumatology in 1963, 11 years before the first board of rheumatology was given by the American Board of Internal Medicine. As a myologist, Dr. Vignos went on to establish the Muscle Clinic at university hospitals to care for both acquired and inherited muscle disorders. Edith Ingalls Vignos passed away in 2005, followed by Paul Vignos in 2010. Dr. Vignos was internationally known for helping children afflicted by muscular diseases to walk, improving countless lives, and advancing research in rheumatology and orthotics. Today, we have in our audience a woman who assisted Dr. Vignos for more than 50 years. Rita Waldheim, could you please raise your hand? There she is. Rita served faithfully as Dr. Vignos' secretary and knows firsthand how his care and compassion improved the lives of children. We are very pleased to have with us here today Paul and Edith's daughter, Karen Sturgis, as well as Dr. Vignos' sister-in-law, Pat, and nephew, Joe. So we're thrilled. We are also pleased to have Dr. Bobby Brown, her husband Steve, and I think her sons didn't make it this afternoon to join the celebration. I'd like to invite Dr. Brown to join me on stage and tell us a bit more about the legacy of the Ingalls family. Now, as she's coming up, let me tell you that Dr. Brown is the granddaughter of Louise and David Ingalls, the niece of Paul and Edith Vignos. She serves as president of the foundation, now in its third generation of honoring the family's legacy, supporting the health and wellness of Cleveland and beyond. Dr. Brown has a PhD in anthropology and biomedical sciences, which seems fitting given her family's support of these causes, stretching back almost 100 years. In addition, she spent more than a dozen years teaching anatomy at the Northeastern Ohio University College of Medicine. Bobby, we're really grateful for your family's support of this professorship, and I'd like to invite you to say a few words.
Hi, and welcome. I'm so honored that all of you came and helped us with this celebration for this chair. As Pamela said, Dr. Vignos and was my uncle. We called him Uncle Vignelli, and his wife was my mother's eldest sister, Aunt Edie. And so for our foundation, which was started by my grandparents, it was not such a difficult thing for us all to agree to support this chair. And each of us, there were 24 of us, cousins of grandchildren of my grandparents, Louise and David S. Ingalls. And so all of us get together twice a year, and not all 24 of us, but a good representative of the family get together to try to decide what things that we would like to support. And University Hospitals and Case Medical School has always been one of the top things that we have cherished and that my grandparents have cherished. And in some of the literature that I found, it's a place that we should always try to support whenever we can. And for Dr. Vignos and my Aunt Edie, they were world travelers. They knew a lot about everything. Uncle Paul Vignos was a scholar. It sounds as though Dr. Medoff is the perfect choice for this chair, because as his career has spanned so many things, so did Dr. Vignos, and he was a scholar, a world traveler. You could talk to him about anatomy, you could talk to him about medicine, you could talk to him about the Mamluk architecture in Egypt and Cairo, and he knew just a little bit about almost everything. So it was fascinating to grow up with him and to talk to him. So thank you all so much.
Thank you, Bobby. Now I'm pleased to introduce to you Dr. Clifford Harding, the Joseph R. Kahn, MD, Professor of Pathology, Chairman of Pathology, and Director of our Medical Scientist Training Program. We're delighted to have him here today to share with us some remarks about the department. Dr. Harding?
Thank you. Well, thank you, and let me start off by adding my welcome to you all for this wonderful ceremony to honor Dr. Medoff on his the award of the Dr. and Mrs. Paul J. Vignos Professorship in Rheumatology, Immunology. I will, and actually I'd like to follow the previous comments about the wide breadth of important contributions. Dr. Medoff has gathered an audience here. I was looking at who is here, and it actually represents a remarkably broad spectrum of departments: physiology, pathology, rheumatology, biology, ophthalmology. I know there are others. So his impact has been wide and deep. I'm going to keep my comments short, though, because Dr. Davis has already given some introduction, and Dr. Kaplan is going to actually introduce Dr. Medoff. I would just like to put Ed into the context of the community and communities that he has been part of. Ed is a rheumatologist, and he is a internationally renowned immunology researcher. Here at Case Western Reserve University in the School of Medicine, the pathology department is the main home of basic immunology research, and as a result, Ed's primary appointment is in pathology, in addition to his appointment and service in rheumatology in the Department of Medicine. And it's important to note that the Department of Pathology has a long and illustrious history in immunology research that includes specifically a series of leaders in the field of complement research. Some of the early members of that, the school of complementology here include Enrique Ecker, who worked on defining the components of the classical complement activation pathway. Louis Pilmer, who Dean Davis mentioned just a minute ago, who was actually Ecker's student, but then went on to become a senior figure in complement research and actually made the major discovery of the alternative pathway of complement activation. And Erwin Liepau, who worked with Pilmer and in his own right then became a leading figure in complement research. And in his memory, we have the School of Medicine's annual Liepau Medical Student Research Day. So this is a long series of internationally renowned figures in the field of complement research, and Dr. Medoff is carrying that tradition forward. Dr. Kaplan is going to introduce him in more detail, but so I don't need to specify the areas of his scientific discoveries, but suffice it to say he has been working on how complement regulates many aspects of human physiology and disease, and really reaching into some very novel areas with important implications for medicine. I'll now introduce Dr. David Kaplan, who will introduce Dr. Medoff.
Dr. Medoff received his undergraduate degree in organic chemistry from Caltech and his 2 doctorate degrees from the University of Southern California. He trained as an internist at USC, Columbia, and UCLA in successive years and was trained as a fellow in rheumatology at Harvard University and the University of Chicago. He stayed at the University of Chicago on faculty, and in his first year gave a rheumatology lecture to the second-year medical school class. It was actually a terrific lecture. I know because I was a second-year medical student at the University of Chicago at that time. And that's a picture of Dr. Medoff from the University of Chicago. And And also, as a medical student, I actually delivered a sample to his immunology diagnostic laboratory that Dean Davis mentioned. I liked his lecture so much that I tried to sign up for a clinical rotation with Dr. Medoff. He must have been popular with my fellow medical students because I did not score a high enough ranking in the lottery system to get the rotation with Ed. Dr. Medoff went on to a faculty position at New York University, and after a few years there, he was recruited to the Department of Pathology at Case Western Reserve University by Dr. Michael Lamb, who was chair of the department at that time. And in fact, Ed, Mike just called me about an hour ago on some other issue, and I told him about this, and he was very excited for you and congratulated you on this. Great honor. I remember that Dr. Lamb was excited about Dr. Medoff's recruitment because he had a high regard for Ed's scientific capabilities. I think it was a fortuitous recruitment for CWRU as well, because we all have had a front row seat to some of the finest, most creative, and most rigorous scientific investigations conducted over the past 30 years. Just before his recruitment to Cleveland, Ed had discovered DAF, or decay accelerating factor, later named CD55. And this is a picture of Ed from his laboratory at NYU where the discovery was made. I think this picture was taken right around the time of the discovery. This seminal discovery led to a variety of biological and biomedical insights that informed advances in the field of complement. DAF is a complement regulatory protein. It decreases the capacity of complement to mediate cytotoxicity against autologous cells. This function is crucial in maintaining the immune function of the complement system. Ed's discovery led to the identification of other intrinsic regulators of the complement system. His discovery also led him to characterize the biochemistry of the unexpected and unusual anchoring of the molecule to the cell surface. This basic scientific study led to his identification of the genetic defect seen in paroxysmal nocturnal hemoglobinuria. It also led to a novel bioengineering capability, the painting of molecules onto the surfaces of cells. Ed continued his investigations of DAF to delineate its role in the pathogenesis of myasthenia gravis, membranous glomerulonephritis, inflammatory bowel disease, peptic ulcer disease, atherosclerosis, and myocardial infarction, among others. More recently, he discovered that immune cells have the capacity to produce complement proteins locally, a finding that violated currently held dogma. These findings led Ed to postulate and subsequently demonstrate that complement components regulate T-cell immunity. This observation, which forms the basis of Ed's current efforts, is also a strikingly novel finding that has upended our understanding of the regulation of cellular immunity. His innovative interpretation of these findings has led him to investigate the important role of complement proteins in receptor-mediated events beyond the immune system. His current studies have important implications for our appreciation of the pathogenesis of cancer and certain neurological diseases. Ed is drawn to address important topics with rigor and creativity. Besides his scientific achievements, Dr. Medoff has an impressive academic legacy. He has been a valuable mentor to a small army of students and fellows. An important aspect of his success has been his dedication to the teaching mission of the university. Ed is a brilliant scientist, a compassionate physician, and a dedicated academician. His work demonstrates profound insights into the biology of disease and ingenious applications to clinical medicine. It is my honor to present Dr. Ed Metoff.
First, let me thank you all for coming and say that I'm very humbled by this great honor and give thanks to the Vignos family, and to Pam, and to Cliff, and for Dave for that wonderful introduction. So I have a little bit of a challenge here, which I hope you'll bear with me. It's a diverse audience, and I have to compact 3 or 4 decades of research into, they tell me 20 minutes, and I hope I don't run over. But also, I thought it would be important to include not just the past, but where we go from here. So it's going to be divided in 2 parts. I'll give 4 examples of the past, and then I'll have a transition slide where we go with here with rheumatology participation and with new research programs. Okay, I'm told that I advanced the slide with this, but oh, sorry, okay. Okay, past and present, as I said, and future and beyond, and that'll be plans and some selected examples.
You need to turn off the other microphone, the one in front of you.
How do I do it? Okay, thank you, Cliff. So I also want to thank Mike Stranick, who helped greatly with this presentation. So I thought I would start with this analogy. If you take your car to a mechanic, you want to know that the mechanic knows how the car works. So one approach that has been a centerpiece is to better treat immune diseases, first understand how immune processes work normally. This is somewhat unusual and a little bit harder to get funding for. So these are pictures which are in the rheumatology book to present manifestations of systemic lupus erythematosus, the facial. The important thing here for going forward is the pathogenesis of the disease has to do with deposition of complexes of antigen and antibody into tissues, in particular, as shown here by the arrow, in this basement membrane in the kidney. So the first question is, this is an area which I started on first, trying to look at abnormalities in lupus patients, but then asking the question, what actually functions to prevent this deposition? So it turns out that red blood cells, which we all know serve the critical purpose of carrying oxygen to all of our tissues, they actually have a receptor called C3b receptor, or otherwise called CR1, on them, the function of which was not known. So what I'm going to show you is that red cells actually have a second function, and this is relevant to lupus because their function is to pick up immune complexes, antigen-antibody complexes, and these designations are for bound complement fragments to them, and then to carry these complexes, transport them to the spleen and liver, but not only to transport them, but to process them so that they can be taken up by those other cells. So this is a step-by-step animation. First, the antibody comes from B cells, which we'll talk about later, and binds to the antigen. Then you have antigen antibody with complement C3b, which is the ligand for that receptor. And then the complex actually binds to the red cells. Now this was sort of unexpected because red cells have very few of these receptors compared to other blood cell elements. But of course, the key here is that red cells outnumber the white cells by 1,000 to 1. So the next step is that while the red cell carries the immune complex, the complement fragment C3b gets converted into another fragment called C3dG. And then the next step is that the red cell takes this to the spleen, and C3DG then is able to actually bind to the very cell which makes the antibody, the B cell. And then this feeds back to amplify the antibody production. The red cell then returns to the circulation to repeat this function as well as carrying oxygen. So the first lesson is red cells have a second immunological function. Okay, now what are the factors needed for red cells to carry this function out? Well, it's the classical pathway components of complement, which are C1, C4, C2, and C3. And what's given down here is actually the percentage of patients who have complete deficiencies of these proteins, genetic deficiencies of any one of these proteins. These are the percentage that actually develop lupus. So this actually is a genetic defect, or 4 genetic defects underlying the development of lupus. Okay, the second example, in addition to clearing antigen antibodies I've just shown you, and in addition to the well-recognized purpose of the immune system to recognize and kill pathogens, the immune system has to do an equally important job, which is to discriminate so as not to attack our own body proteins to prevent self-injury or autoimmunity. So how does this occur? So cells are protected from immune attack by this protein, which stands for decay accelerating factor. So this diagram shows that when complement is activated, it will be able to kill a bacteria, but it will be shielded from damaging self cells. And that's because self cells express the DAF protein, which is diagrammed here. It's got 4 segments in it, and this is its active site. And this is actually the actual structure, which we did by nuclear magnetic resonance. Now, DAF, as indicated by Dave, is anchored to the cell membrane in a special way, which was novel at the time we first described DAF. This is called a GPI anchor, which differs in the way most other proteins are anchored. And the actual in vivo relevance of DAF function, well, okay, so let me show you how DAF works in this following animation here. So this is a self-cell membrane, which has DAF on it. And this would be the classical pathway of complement and the alternative pathway of complement trying to activate on a self-cell. So you can't stop the initial steps, but what DAF does is the minute the complement begins to bind, it kicks off the further progression of the cascade. Okay, now, I showed you the idea that DAF protects self-cells, but what's the actual physiologic proof of that? This is a disease called paroxysmal nocturnal hemoglobinuria. This is a rare but extremely interesting disease. The clinical manifestation is that the patients wake in the morning with reddish type urine. And this is actually urine that I collected from 3 PNH patients when I was in New York. And this is actually their serum. It also shows the red pigmentation. And the red pigmentation is due to hemoglobin, which comes from their red cells. And what happens in this disease, and the reason that the urine is red in the morning, is that they lyse their red cells at night. So many studies showed that this was due to a mutation of a bone marrow progenitor. Progenitor is the cell which gives rise to all of the daughter cells of the hematopoietic system. And it's the loss of DAF that allowed this to happen to red cells to be lysed. So initially, we thought that the defect would be in the gene, which encodes for DAF protein. But studies of the protein and studies at the mRNA level showed that those were normal in PNH patients. So focus was placed on the GPI anchor, and that actually is the site of the mutation. So the second lesson here is what is PNH, and the proof that DAF is important physiologically, because if you don't have it, you would get this disease. So let me show you a little bit about a GPI anchor. So a GPI anchor is added to proteins as they go through an internal structure in the cell called the endoplasmic reticulum. So the protein threads through its normal N-terminal signal called signal 1. But proteins that get GPI anchors have a second signal called signal 2. And the GPI anchor is actually totally pre-assembled and then added en bloc to the end of the protein as it comes out of the ER and heads towards the cell membrane. Now, studying the GPI anchor and understanding it was not only important for DAF because other proteins were shown to be GPI anchored later, and one of significance would be prion protein, which leads to Jacob-Creutzfeldt disease. So in order to find the defect in PNH, one needed to know the steps of GPI anchor assembly. So the first step, I'll just go through, we don't have time to go through them in detail, but just to show them sequentially, then you get the fully assembled GPI anchor. And many people were involved during this period of time in working this pathway out. Now it turns out that in PNH, the mutation actually is in the very first step of the pathway. And the GPI anchor terminology was turned around this way to call PIG. So this enzyme is called PIG-A. And this enzyme is the one that's mutated. And without that enzyme, although you have DAF protein, you can't be expressed because you don't have the GPI anchor. So complement attacks the cells. And that's why you get hemolysis and red urine in the morning. And actually, in the disease, what happens is the mutated cell expands out and grows out of the marrow and tries to take over like a cancer, and it becomes a bigger proportion of the hematopoietic system. So working with the Alexion company, we helped to develop this treatment, eculizumab, anti-C5 antibody, which is not only will totally control PNH, and what's shown here, all these numbers are different PNH patients. This is a paper from another group in New England Journal in 2004. This is hemolysis here on this side, and here the eculizumab is given, and it shows hemolysis totally goes away. It's expensive, like many of the biological compounds. Okay, the third example I'm going to go into is muscle disease, and I thought it very appropriate at this point to show a picture of Dr. Vignos, who was a pioneer in this, and this is one of his papers, a biochemical study of certain muscle constituents in human muscular dystrophy. It's a JCI, which is a high-impact journal, and Dr. Vignos published 68 papers and a text and wrote a book. I made great efforts to find the best picture I could, and I thank Pat for this one. So to transition to muscle disease, another muscle disease is called myasthenia gravis. Now this is a muscle disease in which patients elaborate an antibody against the muscle end plate where the nerve connects to the muscle, and the antibody prevents the transmission to the muscle. So what we're showing here, and I'm going to show this in other slides too, is experiments using a DAF knockout mouse where we've selectively knocked out the DAF gene. So the mouse is otherwise normal, except it doesn't have the DAF gene, another in vivo proof of concept. And I want to thank Feng Lin, who was in the lab at the time and actually made that mouse. So we inject the mouse with an antibody comparable to the antibody that's elaborated by the patient. And we have the wild-type mouse, and we have the DAF knockout mouse. And the next I'm going to show you a movie showing the comparative strength. And the way that you look at the strength of mice is that mice are able to hold on to a cage like this here. If you just hold them up, their paws will reach up to the cage, and they'll hold on. And a wild-type mouse can hold on for more than 10 minutes. And DAF knockout mice will fall off in 8 seconds, which I'll show you in the movie. So this is the DAF knockout, this is the wild-type. Hopefully the movie works. So another in vivo proof of the importance of DAF protein. Now, one of the unusual things about myasthenia gravis is that it starts in the eyes. And up to 15% of patients with myasthenia gravis, the disease just remains in the eyes, and nobody understood why. So work done with an ophthalmology researcher who was previously here, Dr. Porter, using gene array, showed that actually the number of DAF molecules in eye muscles is 7 times lower than in other skeletal muscles. So these muscles are preferentially susceptible to complement attack. So that's why myasthenia gravis, that's the third lesson, why myasthenia gravis starts in the eye and why the eye is more susceptible, and another importance of DAF in muscle disease. And we haven't looked at muscular dystrophy, but I'm interested in doing that. Okay, moving to another topic. Now, the immune system usually keeps complement over in what's called the innate immune system, which is the immediate, nonspecific reacting part of the immune system, as opposed to what's called the adaptive immune system, which involves T cells and B cells, T cells being very important, in particular, well known for AIDS and for viral infections. So the traditional understanding, even in the 2015 textbooks that we teach immunology to, is these are totally separate. So the T cells and adaptive immunology B cells are separate from complement, which is in the immediate, nonspecific immune system. And I have written here wrong. This is a typical diagram you'll see in a textbook of T cell activation. What happens in T cell activation is an antigen, a macrophage-like cell, called an antigen-presenting cell of various types, processes an antigen. This is an area of Dr. Harding. Picks up the antigen and then presents it in what's called an MHC class II complex. And then a T cell, which has the receptor, which sees the two of them together, then gets activated. And this involves other signals called co-stimulatory signals. And this is what causes proliferation of the T cell, so that after a few days, you have enough T cells to fight off the viral infection and to protect against AIDS. So this is the new schematic that actually antigen-presenting cells and T cells themselves make complement. Now we call this local complement as opposed to systemic complement. And it's the earliest event in T cell activation, and I'm showing all these complicated steps, but we get a third signal up here where the APC then makes another cytokine which polarizes the T cell to become a Th1 cell, in this case, IL-12, to make interferon. Okay, how do we prove that this is really true in an in vivo situation? So again, we use a knockout mouse. This time it's a knockout mouse which lacks receptors for activation fragments of complement, the C3a and the C5a. And we have to knock both of them out. So that's the only thing knocked out in this mouse. And we infect the mouse with herpes virus, both mice identically. And what we see after 2 weeks, we see that this is work done by Feng Qi, that in the wild type, the cornea will become opacified. But remarkably, in the double receptor knockout, consistent with the fact that the T cell response, the eye looks normal. And this is because the damage in the eye actually is not due to the virus, but it's due to the immune response, the T cell response against the virus. And this mouse, who doesn't have the receptors for these complement activation fragments, can't make a T cell response against the virus. Okay, now in T cell immunology, there's a subset of cells which are the master regulators, and this is important for autoimmunity, and we'll come back to this later in the context of not only lupus, but other autoimmune diseases, including multiple sclerosis. So these cells, this subgroup of cells is called T regulatory cells, and they're characterized by this, this is a transcription factor called FOXP3. It's not important to remember the name of this, but only the fact that these cells, which make up 5% or so of the T cells, they are the cells which are critical to confer tolerance against our self-antigens and to prevent autoimmunity. And as I indicated, they're characterized by the expression of this transcription factor. And they have these other proteins on them, which people use as markers. Okay, now, the simple story here, what I always like to do is take all of this complexity and try to get a simpler story. I work under the concept that when you get a simple story, then you know it's right. Hopefully, usually. So here's an antigen-presenting cell, here's a naive T cell. So when they both make complement and they both elaborate these activation fragments, the naive T cell will become a T effector cell. And there's different types of T effector cells. We don't have to go into that. The important point is when the two partners make the complement, when they're interacting, you get the T effector cell. When they don't have complement activation, when local complement is not produced by the partners and C3a, C5a not generated, the cell defaults and becomes the Treg. So this is the normal default mechanism, so that always in our lymph nodes, we don't have this type of reaction going on, and we're making T regulatory cells, and we're not getting self-attack. Okay, now this knowledge is very important for treatment, because this is a comparison of the suppressive strength of T regulatory cells made by blocking the complement signaling, as opposed to the way that it's conventionally made by other people by adding a cytokine called TGF-beta. So they're 16 times more suppressive. And this is a very important point, that ones made by the conventional methods lose their FOXP3 and lose their suppression, whereas ones made this way are more stable out to 4 months. And this is work by Mike Strainik, which is just being submitted. So, as I mentioned, moving to another autoimmune disease, multiple sclerosis. This is well known to be T cell mediated. What that means is that there's a defect here in the immune system in that it's not functioning, as I showed in the third or fourth slide, to prevent attack on our own cells, but it's actually attacking our own cells. It's actually attacking the myelin on the nerve sheaths. So here, the experimental model is that we just use a wild-type mouse. And this experimental model is called experimental autoimmune encephalitis. So what you do is you take a component of the nerve sheath called myelin oligodendrocyte protein, MOG, and you immunize the mouse with it. And when you do that, the mouse will become sick. So this is the clinical score here, 0 being normal and 3, 4 being worse. And this is the weight of the mouse. As the mouse gets sick, it can't reach the food up on top, so it loses weight. So the black line shows what happens to the mouse after you induce this EAE in them, which simulates multiple sclerosis. And it shows that the mouse gets sick, shown here, and the mouse loses weight. But if you give them these Tregs made by receptor antagonism, the clinical score goes down and the weight returns, and we get a happy mouse. Now, is this translatable to humans? Okay, so here what we do, and this was done by Dr. Westbrook and Devereaux, who are in the back, who provided these patients' blood cells, and we isolate the T cells from the patient's blood cells, and then we can treat them with this methodology to block the complement signaling to make FOXP3 positive cells, and then we can sort those, and we can examine their suppressive capacity, as opposed to the conventional method with TGF-beta. And this is suppression at different ratios of effector to Tregs. So you have 8 effectors to 1 Treg going to, in this direction, 1 effector to 8 Tregs. And you see that the ones made with TGF-beta don't work. So you can't make good Tregs out of the blood cells from MS patients in the conventional manner. But if you take the blood from MS patients and you treat them with the receptor antagonists or with antibodies against the ligands, you can make suppressive. So these are the 2 treatments which are overlapping here. So in all patients studied, there's 3 shown here and 5 or 6 more which we have done, which give, in all cases, the same results. You can make Tregs, which will suppress the effector cells. So we got this one out of order here. Oh wait, I'm sorry. My fault. So before I talk about the treatment, let me come back to lupus. So the first diagram about the immune complex clearance and the C3DG on the red cells was that the C3DG carries the antigen antibody complex to the spleen, and the cell it reacts with is the B cell. And here, what we show is that if we immunize a wild-type mouse versus this double receptor knockout mouse, this is a B cell response, and this is just an immunization with ovalbumin, which is just a sample protein. That in the not only the double receptor, not only the complement signaling controls the T cell response, but it also controls the B cell response, because the double receptor knockout mouse can't make antibody. And in opposite situation, the DAF knockout mouse, which allows more local complement signaling as opposed to this one, which has no complement signaling, makes much more. And I point out that this is at 7 days as opposed to 13 days here. So the local complement signaling controls not only the T cell response, but also the B cell response. So this is actually a mouse model of lupus. I understand I was switching from multiple sclerosis to lupus, but the reason being that they're both autoimmune diseases, T cells first, B cells second, and then the treatment I'm going to give next. So this is a lupus model, a lupus mouse model, where we could take the wild-type mouse or the double receptor knockout mouse, and if you treat it with this agent called pristane, the wild-type mouse will develop lupus after 5 months. And this is shown up here. This is the diaphragm of the wild-type diaphragms, and this is the spleen. And Howard Meyerson helped with this. So this is, the wild-type mouse gets these typical lymphoid follicles. These are lymphocytes, T cells and B cells in here, in the wild-type mouse in the lupus model. And gets this is called follicular hyperplasia, where the number of B cells in the spleen is greatly increasing as compared to the normal architecture of the B cells. And the identical treatment with this double receptor knockout mouse, you see no disease and no change in the spleen. And the hallmark of lupus is anti-DNA antibody, and as opposed to the wild-type mouse, which makes the DNA antibody simulating the human situation, the double receptor knockout mouse does not. So this then puts us in the position, both for lupus and for multiple sclerosis, to do the following thing. Take the cells out from the patient. In both cases, isolate the T cells and block the complement signaling into the T cells. Then sort later for the Tregs, which I showed you in a previous slide. By this treatment, we've been able to energize them to become suppressive, and then give them back to the same lupus patient or same MS patient. And then this would inhibit the autoimmune response, and the patient theoretically get better. So we have a new grant for this in lupus, and if this study works out, we can then go on to a clinical study in lupus or multiple sclerosis. Okay, moving to the second half. Immunology 2016 going forward. So the centerpiece of this is to develop new immunology interconnections. So 2 objectives: to to enhance and continue to enhance research in rheumatology. The second one, as Dean Davis mentioned, is this new initiative in integrated basic immunology, which involves cross-disciplinary scientists who work across different disciplines. The student program, for which I have 7 so far, and some of them are here, and then an initiative to start exploratory work in as yet non-immunology relevant areas such as muscular dystrophy or other eye diseases. And I'm going to end the talk here by showing you 3 examples. All right, now in cancer, it's well known by all cancer immunologists and all cancer people that immune cells are important to have immune surveillance and to inhibit cancer growth, and hopefully in most cases prevent the tumor from developing. But what's unknown is that immune proteins in the tumor itself, specifically complement proteins, regulate the cancer growth and progression. And I'll give you 2 examples of this. So this was done with Dr. Sheeman in the Cancer Center. And this is a 4T1 mouse breast cancer. This is the more aggressive type of, it's called a triple negative breast cancer. So growing it in culture, in the absence of any immune cells, no immune cells present, in studying, this is the number of cells growing over time, just counting them. So you see the rapid growth and progression of this cancer. But if you block the receptor signaling, because the cancer itself, just like immune cells, makes the local complement, which autocrine signals back through those receptors, which is essential which participates in the cancer growth. And this is a 3-dimensional picture of the cancer uninhibited and the cancer in which the receptor signaling is blocked. The next one is Dr. Daniel Poor, who's in the audience, studies prostate cancer. So this is prostate cancer, and this is the growth of this particular prostate cancer in vitro, and in vitro in this particular sense, and in the absence of the receptor antagonist and in the presence of the receptor antagonist. But the point being made about this slide is that for cancers to grow, they need to have a cancer-associated blood supply to grow with them, which is called angiogenesis. So the receptor antagonism is not only fundamental to the receptor signaling is not only fundamental to the cancer growth, but it's critical to the growth of the blood supply for the cancer. And this shows The brown here are staining for blood cells, and this shows the blood cells associated with the cancer as compared in the double receptor knockout mouse or in the receptor antagonized mouse. So when you receptor antagonize, you block both the cancer and you block its cancer-associated angiogenesis. Okay, wound repair. And this is work done with Mario Cohen in ophthalmology. Complement proteins promote corneal regeneration. So what's done here, this is mouse eyes, and Mario figured out how the little tiny eyes, the size of a pea, of how to make a reproducible size lesion on the surface, which only goes through the corneal cells. And then if you stain with this stain fluorescein, the fluorescein will only be picked up in the wounded area, so it's green. So this is a wild type mouse, and then 6 hours later, you see that the wound hasn't progressed very much. But if you do this in a DAF knockout mouse, in 6 hours, the wound is almost completely healed. So we made an antibody, a mouse anti-mouse DAF antibody. And the experiment in progress is to develop this anti-DAF antibody in the wild type, and this is a human eye, so that eventually we could move to a drop, which could be put in for patients that would come in with major defects in the corneal epithelium. Nerve regeneration. So when we're talking about integrative immunology, moving into these kinds of areas which are generally thought to be other disciplines. So this, I think this is the last example I'm going to show. So we have here the wild type in the DAF knockout mouse, and we harvest the cerebellum from a 13-day embryo. And we treat this, which is done with Dr. Zhu, with nerve growth factor. And then the black lines here are neurites, which are the new nerve outgrowths. So in the wild type mouse, it's 6.5 per millimeter. In the DAF knockout mouse, they're growing much faster, 45 per cubic millimeter, and this is diagrammed here. So the idea here is that somebody that has a nerve damage, that it possibly may help if we would inject anti-DAF in there, which would stimulate which would promote the nerve growth. And actually, we had some data of that in our EAE experiments, which was done by Jimbo, who's in the back. So just to finish, all I did was sit in the office and write papers and write grants. These are the people that did all the work. Thank you.
Ed, please stay on the stage, and I can invite Dr. Brown to return to the stage, please. Thank you very much for that very enlightening presentation. We are delighted, Dr. Medoff, to honor you this afternoon as the inaugural Dr. and Mrs. Paul J. Vignos Professor in Rheumatology and Immunology. And Dr. Brown, the entire School of Medicine community is grateful to
Thank you very much.
H. and David S. Ingalls Foundation for making this important professorship possible. Ladies and gentlemen, it is our privilege to present two beautiful chairs this afternoon, one to our inaugural professor and another to the Ingalls Foundation, who made this professorship possible. Please have a seat in your chairs. And this is yours.
Oh, okay, great. Thank you.